welcome to our third vodcast in our regulation domain. In this one, we're going to look at osmoregulation and the ways that plants and animals both balance water content. So in this one, big things we need to understand. How do organisms regulate their internal conditions, and how is that regulation accomplished? Because between animals and plants, very, very different situation. Okay? So without further ado, we're going to start with plants on this one. So plants, due to their inability to move, plants are essentially osmoconformers. Okay, so which makes sense. They can't get up and walk around. So, however, internal osmolarity kind of has serious consequences, which means if you don't have the right osmotic pressure, you're going to go and you're going to wilt. So to review that idea of diffusion and osmosis that we've covered so extensively, okay, the ideas of water flow, when you have a hyper versus hypotonic solution, okay, the relationship between how solute concentration and water concentration exists, right? So if I have a side that has a low solute concentration, high water concentration, I'm hypoosmotic, okay, or what you could say is hypotonic, versus the other side, which has a high solute concentration, hypertonic, and lower water. So osmolarity is that total solute concentration expressed as moles of solute per liter of solution. Okay, so moles per liter. So for example, human blood is about 300 milliosmoles per liter. Seawater is about 1,000. Okay, so blood's obviously kind of more hypoosmotic than seawater. So animals. Okay, here's osmoconformers versus osmoregulators. Some animals that remain isoosmotic within their surroundings, no matter what situation they're in, you know, they can kind of handle that that existence. This would be something like organisms that live in salt water versus regulators that have to constantly be doing something to regulate their internal osmolarity. So, for example, you got your jellyfish, right? So your jellyfish, the cnidarian group, they're osmoregulators. They're going to, you know, have to be able to change their osmolarity because of their huge surrounding of seawater. Otherwise, they would be in big trouble. Okay, and then osmoregulators, that they can live in many different environments. Okay, marine fish versus freshwater fish, how they handle that kind of exchange as they're bringing water in and out. So these adaptations allow for them to be able to regulate how much water they have, when they have it, when they can exchange for different materials in those different situations. So they're going to have to be able to either take in greater amounts of water, excrete less, or vice versa. So here we have some fish. Okay, so salmon are really good at tolerating both kinds, and they can hang out in freshwater as well as saltwater. You know, since most animals can only have really thin windows of being able to tolerate that. Okay, and so urylene is what we call it, and then stenohaline. So euryhaline and stenohaline are the two terms we use. Okay, tardigrades. Okay, these are funky little critters. They live in environments that are only hydrated very, very temporarily. So little drops of water, seasonal ponds, things that come and go often with the amount of water that's there. So in order for them to survive in, you know, very, very dry environments versus very, very wet environments, okay, they have to be able to hang out in a very dormant, almost dehydrated state. And they can do this for decades. Okay, so there you have one that's hydrated versus one that's not. So terrestrial animals consistently are going to lose their water to the atmosphere, okay, and so that's why we have to be able to constantly input greater amounts of water to relay that. So something like a kangaroo rat that you have there, their water metabolism, they get most of that from metabolism, their gain a little bit less from food. Okay, their loss is mostly through evaporation, through the urine, through their feces. Whereas for us, okay, we get more of our water ingested through food, for metabolism, right? So breaking down, we release water molecules, or ingested via as liquid. Our water loss comes in the form of mostly urine, some evaporation, okay, and then the smallest amount in our feces. So that comparison of daily water balance in the rat, which is a desert animal, just keep that in mind, and the human, you know, between human, even though we're both mammals, we both have kidneys, a little bit different in how we handle that water. Osmoregulation, osmolarity excretion. Animals regulate that osmolarity, controlling the amount of solute they retain in their body fluids. So we do this through that mechanism of filtering. If it's a filtering system, so we intake water, we excrete fluid, various other dissolved solutes, and the majority of those animals, that they control their internal osmolarity this way. We maintain what we need, we get rid of what we don't need. So all of these excretory systems depend on these specialized transport epithelia to move those specific solutes either in or out. So here they go. So just a couple of ideas. Production of these secretions in the nasal glands of the albatross allow it to survive drinking seawater. We know if we were to drink seawater, it would kill us for obvious reasons, if we constantly drank it because it's too salty and we couldn't be able to contain that. However, albatross can do that because they have, okay, these glands, okay, that are able to capture the water out of it while excreting the salt ions, and then it drips out the end of their nose. Kind of weird, but it is the way it works. So you can see how the salt movement is counterflow to the blood flow. Okay, and what that does by having that counterflow system, okay, it's constantly keeping a higher concentration of salt, okay, away from the blood, moving that salt out of the blood into where it's going to get secreted out. And so that way it's always higher, and that way it'll always push that higher concentration out if you have that counterflow system, which is pretty cool. So the nitrogenous wastes that are created as well as a waste product. So in that efforts of maintaining water, but however you've got to get rid of wastes, okay, these are produced often in the form of a nitrogen waste, and this is where you're going to get your ammonia, your urea, which is your urine, uric acid. Okay, the most toxic is the ammonia. It's only produced by aquatic animals. Okay, as we break down proteins, as we metabolize, we build up these nitrogen groups that, we have a lot of amine groups, right? Urea is formed when you combine ammonia with carbon dioxide. Not really as toxic unless it builds up in huge amounts of concentrations. Okay, and it's released as we get rid of the water. Uric acid is least soluble, excreted with the least amount of water, and way more energy expensive, okay, to produce. So the form that animals take when they're producing nitrogenous wastes, you can connect that in with phylogeny, where it lives. Okay, there is a connection and a piece that way. So here's what happens when we break down proteins versus breaking down nucleic acids. We get those amine groups. The aquatic animals are going to produce the ammonia as their nitrogen waste product. Mammals like us, we're going to produce it as urea. Amphibians will, bony fish will. Your cartilage critters like sharks. Then you get some reptiles with some birds, insects, and land snails, of all things, will produce uric acid. All right, and then you can see here, bird guano, commercially viable, or not even viable, valuable source of nitrogen, okay? And what we're talking about is bird poo, okay? Same thing with like bat poo, okay? So they call it guano, high nitrogen content. So systems that focus on excretory. We're going to focus a little bit here on some different groups and how they deal with this. The main gig with this is being able to create a process where you get filtration, reabsorption, secretion, and then excretion. Okay, that's kind of the mechanism steps that occur. All of the systems are going to involve these four major processes. Filtration, initially getting the movement of fluid and solutes from the body into the system. We're going to create a filtrate. We're going to shove it out. Then we're going to realize, hey, we just shoved out more stuff than what we need. We need to reabsorb some of the things that we don't want to get rid of. Okay, we're doing that. Then it's going to continue through, and we're going to secrete out some more of the stuff that we don't need to keep. And then finally, literally take that modified filtrate and ixnay it out of the body. So all of these happen through a system and series of very specific tubules that are going to be created to help collect this filtrate. Okay, so protonephridia, okay, like your planaria, okay, that's what these are, okay. Their interstitial fluid, which is the fluid that wraps around the inside of them, right, will move to the protonephridia, okay. The filtrate gets produced through the action of ciliated flame bulbs in the cells, like you can see here. So the filtrate comes through those tubes. That bulb then is going to deal with grabbing that, okay, and then shoving it out, okay? That's how that works. Metanephridia, this is in your annelids, okay? They produce from their coleum, which is kind of like their kind of body region, okay, moves then in what we call the metanephridium. So these are things like your worms. So reabsorption and secretion accomplish by transport epithelial that are going to line the border of that area, okay, with a capillary network that's going to then filtrate the blood out. Stored urine gets excreted through external openings. So they have like tiny little holes then that kind of send it out instead of collecting it into like a major bladder system and then going out. So each section has kind of its own one of these. Then in arthropods, which are your bugs, filtrates are going to move from the hemolymph, which is like, it's kind of like their blood. They don't really have like vessels per se. They kind of body wash their blood through them. And through that, these tubules are going to grab the filtrate combined with their undigested food and eliminate it throughout their rectum. So they're going to then shove it out that way. So that's how they get rid of it. Now in vertebrates, we have a little bit more kind of crazy system, but nonetheless does the same idea. Again, it's all about keeping what you want, getting rid of what you don't want. For this, we use our kidneys, okay, and so do all vertebrates. Okay, we have two of them, okay, it sits down for us in our lower back. The filtrate comes through the kidneys, urine gets collected, okay, through a system of tubes called, basically tiny little tubes that then branch off into the ureter, which then collects into the bladder, which then gets excreted through the urethra. That happens whether you're a boy or a girl. So in the kidney is broken down to a really, really neat kind of structure. It's a organ that has tons of tubules that exist there, okay? And with the kidney, we can look at two particular parts. There's the renal cortex. So anytime you hear the word renal, you're talking about the kidney. Renal cortex, renal medulla. The system of tubules, which we call nephrons, run both through the cortex, that outer layer, and run down into the medulla, which is your inner layer. And this is the organ that's responsible for the filtration. Our blood gets cleaned out, water conserved, water excreted if it needs to be, through our kidneys, okay? And these nephrons, our kidneys will contain around about 1 million of these nephrons in them, which is just crazy cool. So in situations where kidneys don't work right anymore and they're incapable of cleaning the blood, you don't want to go toxic and have accumulations of these nitrogen wastes and various other things that you need to get rid of. We have to go through dialysis as a mechanism. So a person sits there, okay, they do the same thing the machine does the kidneys do, so your blood runs out of you through the machine, which has a series of semi-permeable tubules that are tubes, just like what we used when we do our diffusion labs. Okay, but it's pretty time consuming and you have to go a couple times a week in order to keep up with it. So your kidneys are constantly doing it. I think that's probably why we have two. So if one, kind of craps out, you've got the other one. So let's look a little more closely specifically at the nephron. So again, that we have over millions of these things. This interface happens between the circulatory system and the excretory. The circulatory system is going to send a group of capillaries in that are going to wrap around all these tubules and intertwine. Okay, so big aspects of this that you're going to need to be aware of: the glomerulus, the proximal tubule, loop of Henle, distal tubule, and collecting duct. So let's look a little closer here at the organization of the nephron. Your capillary system comes in. You have the glomerulus. This is like this ball, okay, of vessels that come in that are bringing in the blood. Wrapped around it is called Bowman's capsule, okay, and this is the first system of tubules. In the first part, which is filtration, okay, we are going to ram out a bunch of materials into what's called the proximal tubule. It's the closest one to Bowman's capsule. Proximal tubule starts to send the material down that then heads down what we call the descending limb, loops around the bottom, which is the loop of Henle, comes back up the ascending limb, get it descending down, ascending up, and then heads into the distal tubule where the majority of the stuff I'm getting rid of gets then sent in a collecting duct all the way out to the bladder. As it's passing through at various points in time, it is going through secretion, reabsorption, and heading out. And there's going to be various pressure components that exist to cause gradient changes that are going to either take in water, excrete salt, ions and all of that stuff. But at the same time, in different locations as well, there's different permeabilities that exist. All right, so while you're chewing on that, let's take a second here and talk about what actually happens in this. We're going to simplify it down. So filtrate comes in through the cortex, right? So as that stuff is shoved out, okay, there's things that we say, okay, whoa, we don't want to keep, things that we want to pull in. So we're shoving into it all kinds of stuff, but we want to pull back nutrients, some salts, potassium, water, okay, but we want to pull in as well, you know, the nitrogen compounds. Those all start to collect. As we head down the loop of Henle, down the descending side, okay, that's where you're going to get permeability to water, and there's going to be a higher water concentration in the loop than there is out. So it's going to cause water to be reconserved. As it's going down, it's getting more and more concentrated because more and more water, okay, is being taken out. So you get a really concentrated, most concentrated at the bottom of the loop. As we head up the ascending, we're no longer permeable to water, we are permeable to salt. Okay, and we want to get rid of salts. So now that high concentration of salt gets pulled out, and it becomes now less concentrated as it goes up. And then in the distal tubule, there's some re-exchange of some materials, and then we head down the collecting duct, okay, where we're going to come down, grabbing that, bye-bye, waste. So there it is, okay? So by actively transporting, and here's the thing, we're going to have some active transport, or we're going to have some other things. So we're going to force some things, and we're going to change it. Okay, this change happens because of the change in the osmolarity of the filtrate solution. The nephron's going to create a hyperosmotic environment leading to passive transport of water from the filtrate, production of a hyperosmotic concentrated urine. Okay, two solute model. So take a second. I want you to look at this a little bit closer, where you're going to see the varying how the concentrations change as it travels through. So if it starts at around 300, right, as it's going down, if I'm pulling water out, it's becoming more and more and more and more solute concentration. Notice, okay, all passive, so all diffusion. We whoop around at 1200, we come up. Now we've got passive transport of the sodium because it's less concentrated, all the while pulling the urea through. It's all about collecting that. As it comes up through, we grab more. Now we start actively taking salt out. Now we're down to 100. Okay, we hit the collecting duct, and that's where it starts to really change, okay, based on what is needed by the body. All right, so controlling this, how is this actually controlled? Controlled through hormones. Okay, first hormone is called antidiuretic hormone, or ADH. So when the blood osmolarity increases, pituitary gland is going to, again, remember that pituitary gland right between the eyes there. Okay, that pituitary gland is going to release ADH, and as ADH gets released, it acts at the kidney, okay, and that's going to increase water absorption in the collecting duct of the nephron, okay? This is also going to trigger thirst response, okay? So not only that, so it goes to the collecting duct, okay? Water is reabsorbed, okay, to help maintain homeostasis, right? The stimulus, the increase in blood osmolarity, okay, which is your increase in, you know, that solute concentration. So if you've been sweating a lot, goes back up to the hypothalamus, which is an area of the brain just below the pituitary, triggering then the release of ADH. Okay, and then it all kind of goes over again. So this affects, you know, the effects of these decrease the osmolarity of the water. Okay, so once you get that thirst response, you're going to drink more water, reducing blood osmolarity. That goes down. Oh, I've got a lot of water in me. Oh, signal back in. Okay, I want to conserve, you know, reabsorb water, you know, from the collecting duct. So that's ADH. Here's how the receptor works at the cell level. So ADH, again, as a hormone, acts at a cellular receptor on the cell membrane. It sends off a signal of cyclic AMP second messenger system, okay, helping to signal formation of molecules that are going to cause exocytosis and then pull water through, okay, and water comes in and back into the body. So that's cellular response in the collecting ducts, leading to an increase in the number of aquaporins in cell membranes. So an aquaporin is what water goes through, increasing water reabsorption. So it's making you retain water, not get rid of it. That's its job. It's antidiuretic. Then you have the RAAS system when you have a sudden decrease in blood pressure near the nephron. You get this the juxtaglomerulus apparatus. That's that region of the nephron where all this happens. Releases the hormone renin. Okay, this goes with renin, angiotensin 1, angiotensin 2. Okay, that's the system. So the renin that's created results because you convert angiotensin to angiotensin 2. These are all, again, hormones, okay, that are produced, okay, that are going to act at the adrenal gland. Now, the adrenal gland sits right on top of the kidney. Kind of convenient that it sits there. The adrenal gland ultimately produces the steroid hormone aldosterone, which then triggers an increase in the absorption of sodium ions, okay? So for this one, you're pulling in and making sure you reabsorb sodium ions, okay, to increase pressure, which then helps to maintain, pull in more water, get more blood volume, as a result, higher pressure. So if you have a drop in pressure, that's what happens. So there's that, okay? So kind of take a second to kind of get your, mechanism in terms of what's going on. What turns it on, how the homeostasis is, you get blood pressure volume change. So you get low blood pressure that signals this to happen, okay? And all of that happens. So adaptations for this, okay? So here's some examples. So vampire bats, okay, they feed at night, large animals, okay? Drinks lots of blood per feeding, right? So to compensate for huge fluid influxes, the vampire bat will excrete up to 25% of its body mass in urine per hour. That is huge amount. So this adaptation is, because that's pretty much its diet, right, is a liquid diet. So as it's drinking that in, instead of having an over hydration and influx of fluid that it can't handle, as it's doing this, it's excreting out water at the same time. Okay, so it's eating and peeing all at the same time. So there you have it, osmoregulation. So from this, make sure you can explain why the organisms need to regulate their internal osmolarity. Why is that important for functioning? Similarities and differences in the osmoregulatory nitrogenous waste excretion systems in different animal lineages. Structure and function of mammalian excretory systems. We are going to focus on that. So as far as a mammalian point of view, so in general, you should be able to know nephron specificity and how hormone controls do that. So you may get, in fact, that the excretory as your system when we do our physiological project. So there you have it. Have a good night, and we'll catch you later. Bye.